Thanks very much, Rob, and thanks, Viv, for the opportunity to speak, sort of, at Boston. Today is International Day of the Dog, apparently, at least according to Twitter. So I thought, Xander, come on. We're going to introduce Xander here. Hey, buddy. Hi. This is Xander. You'll see him again in the talk, but in the meantime, go away, buddy. So I'm going to start my screen sharing here. Give me a second. And hopefully this works for everybody.
That looks perfect.
Awesome. Thanks, Viv. Okay, so I'm going to talk about awake behaving functional imaging of the marmoset at 9.4T with a little bit of human, a little bit of rodent, and a little bit of macaque thrown in for good measure. So the work I'm going to talk about is actually mostly done by extremely productive postdoc David Schaefer, who has just recently moved after the border kind of opened to Pittsburgh to work with Afonso Silva. And David was a postdoc with Stefan Everling and myself for the last many years. And we're really quite sad to see David go, although our grant budgets are quite happy because Stefan reminded me this morning that he spent something like $40,000 on publication charges for David's rather prolific work. So the primate stuff has been a product of working with Stefan and David for many years. Stefan has been my colleague for a very long time. And the MRI advances I'm going to talk about are courtesy of Joe Gaddy, Alex Lee, Martin Klassen, and Kyle Gilbert, and I'll talk about those a bit. So, you know, when fMRI first started, it was really sort of a macro scale mapping tool, very much blobology. And basically, everybody rushed to map areas of the brain that were activated by various things, including whether you were a Democrat or Republican, I suppose. And as the technique got better, as the spatial and temporal resolution got better, people began to look at the connections between these areas. And this really heralded the era of network neuroscience. Now, in the language of graph theory, nodes, such as London and Toronto here, centers of activation, are connected by edges, in this case, roads. We tend to view these as static, but the strength of the connections can vary in time over seconds, and the connections themselves can vary over millennia when you examine evolution. And of course, within a node, there is activity at the mesoscopic level. So this is an example of mesoscopic activity. So here's a connection that existed 143 years ago between Boston and London, Ontario. So in London, we have the oldest ballpark in the world. Back in May of 1877, the National League champion Boston Red Stockings beat our hometown team 7-6 to win a championship that year. You may not recognize the Boston Red Stockings because I know Martinos has people from all over the world. They went on to become the Milwaukee Braves, and eventually they became the Atlanta Braves, who are still on top of the NL East, I think, at this point. So the point is that nodes and edges can actually move over time and are often evolutionarily driven. So in our lab in London, we have three high field and ultra high field MRI systems. We had the very first Prisma Fit in the world. We had the very first Siemens MRI Plus, which is essentially the Teradot upgrade for the Step 2 platform. And we have a 9.4T, and I'm going to talk about data from all three of these. So just to give you some of the geography, our 7T lives over here, our 3T lives over here, and our 9.4T lives over here. This is the University Hospital, which we're physically connected to. This is the Robarts Research Institute, and the rest of campus lies primarily above this. That's not a very pretty picture. Western's actually a very beautiful campus. This is what Western looks like in a couple of more months, basically. This is our magnet pod, as well as the Brain and Mind Institute, where many of my colleagues live. So when you're thinking about model systems in neuroscience, you usually think about how similar to humans they are in terms of either their genes or their proteins or their receptors or their circuits or their behavior. And the model you pick really depends on which one of these things you're trying to emulate. And today I'm going to consider the mesoscale and the macroscale organization of the brain in choosing a model, and that model in particular will be the marmoset. So macaques have been used in neuroscience for many, many decades, and they have a lot of advantages, including the fact that they have folding patterns that are quite similar to humans. But they have many disadvantages in terms of exploratory approaches. For example, brain areas are very difficult to access because many of them lie deep in a sulcus, such as the frontal eye fields. They require extensive training if you want to use them in awake mode. And there are problems trying to get behavioral effects with things like optogenetic stimulation, for example, likely because the number of cells is so much larger than in the mouse. The marmoset, on the other hand, has a lissencephalic cortex, and that makes it kind of ideal for electrophysiology, optical imaging, and other pseudo-invasive techniques, because the entire cortex is pretty much visible. They're much smaller than macaques, so they're not trying to bite your head off all the time. There are transgenic marmoset models of a number of diseases now. They're very highly social, so they mimic aspects of the human condition, and they are much easier to breed. So why marmosets? Well, marmosets have in particular a granular frontal cortex. And this is important because we think pyramidal cells in granular prefrontal cortex in humans and in primates have sort of a disproportionately high number of spiny processes. And these likely endow these neurons with pretty sophisticated computational abilities, and that enables things like complex cognition and complex social behaviors. So this is a marmoset doing a task, in a touchscreen task that Stefan Everling built. So what I'm going to talk about is the story of our journey to build sort of a super marmoset imager and look at these homologies between humans and marmosets and macaques and rodents. We'll start off with the anesthetized animals, and then we'll move on to the awake animals. So I'm an MRI physicist in addition to being a neuroscientist, and so of course all stories start with RF coils. So our earliest work was inspired by Afonso Silva, and we started developing marmoset imaging methods 6 or 7 years ago with a 15-channel receive coil and a 2-channel transmit coil on our Agilent system with a very small bore gradient coil. So this limited our ability to do anything awake at the time. But with the anesthetized animals, Mariam Garamani and Matt Hutchison were able to do resting state fMRI in the marmoset at 9.4, in the macaque at 7, and humans at 7. And it was really quite striking how many of the networks were consistent between marmosets and macaques, as well as marmosets and humans. And that included visual networks, default mode network down here, salience networks, and so on. Actually, Matt's an example of somebody who's also a dynamic connection. Matt actually moved from here to do a postdoc at Randy Buckner, and then gave up academic life to go work at Biogen, and he's built a really beautiful house. So for all you trainees out there, a lot of options available. You can take these surface maps, and of course you can put them on flat maps and look at the detailed cytoarchitectonics. Then a few years later, we started to examine the homologies between rodent models and marmosets. And to do that and to make really robust comparisons, we had to again develop the hardware that would allow us to do this between rodents and marmosets. So we developed a number of sort of 3D printed holders and RF coils. This is an 8-channel RF coil for the marmoset, complete with ear bars with built-in fiducial markers, eye bars, and so on, basically mimicking a stereotaxic setup. We did this for mice, that's ABC, and for rats, DEF. And all this stuff was made open source. And you might think, you know, if you're doing anesthetized animals, why bother with eye bars and tooth bars and ear bars? Well, without ear bars, even in an anesthetized animal over a period of 15 minutes or so, you can see a lot of motion in X, Y, and Z. This is translational motion and this is rotational motion. But with ear bars, you actually substantially reduce that. This drift that you see in the Z direction is actually primarily gradient heating, which causes B naught to shift. If you look at X and Y, the translations are well under 50 microns. So that's quite important for doing these kinds of good comparisons. As I mentioned, we made this open source, and if you let engineers design everything, you end up with contraptions that aren't actually usable. So our vet techs actually had an enormous amount of input into this to make them actually practical. And when you do this, you can make nice EPI and T2 images. The kinds of things we developed actually give us resolutions that Matt Glasser and David Van Essen like to say respect the cortex. That is, they give us 2 to 3 voxels across the gray matter in each species. So you need appropriately matched resolutions, essentially. We do fMRI at 300 microns isotropic in the mouse, 400 in the rat, and 500 in the marmoset, for example. And you get very nice looking EPIs. With this, you can actually do tractography at the mesoscopic scale. These are 4 different animals, the cingulum, the uniculate, superior longitudinal fasciculus, and so forth. These are done at 600 microns, but I'm really not going to talk about DTI today, but just to show you this can be done. What I am going to talk about a lot is the idea of hierarchical clustering to reveal intrinsic functional architecture, starting from resting state fMRI. So the idea is you take a region, you look at all the individual time courses, and you cluster them into groups that covary roughly the same way, that are constrained by Euclidean distance. And that gives you clusters such as these, 3 clusters from these 9 time courses. And you can overlay these clusters spatially onto anatomic or cytoarchitectonic maps. And so David did this in a paper that was published in Journal of Neuroscience, in which he looked at a variety of different cluster sizes in the marmoset in the lateral frontal cortex, using many runs in many marmosets. And he concluded that 7 different clusters within the LFC was a pretty good representation, and that the boundaries of these clusters overlapped quite nicely with the known cytoarchitectonic areas. You can keep going in terms of the number of clusters, but if you do a silhouette analysis, it actually turns out that 7 is a pretty good place to stop. The important point about that is really that you can see that the marmoset lateral frontal cortex is differentiated just like the macaque and the human. And it's actually very hard, if not impossible, to find this differentiation in the rodent brain. You can use each of these clusters as a seed and examine where they connect cortically. So this is a map of where cluster 1 connects. Here, right in the frontal cortex is cluster 6, for example. So of course, the hottest area is itself, but you see where else it connects to. And so by using each cluster as a seed, you can examine the cortical connectivity of those areas. And just to show that we're not cortical chauvinists, you can actually do this at subcluster connectivity as well, looking at the subcluster connectivity from the lateral frontal cortex. So you see the pulmonar, you see the colliculus, you see lots of areas in the thalamus. So you can do the same trick in the medial frontal cortex. So using the hardware that we built, you can look at this across species, so in the rat and the marmoset. And then David actually pulled 8 subjects at random from the 3T HCP data. And he did the same exercise of varying clusters and settled on 4 for reasons you'll see in a second. But you can see across the rat, the marmoset, and humans, this quite well-preserved architecture. If you look at that in more detail, comparing it to the Paxinos atlas, for example, it actually looks really promising. So you can see the medial frontal cortex. The first cluster sits in area 25 in all 3 species. The last cluster kind of covers areas 24 and areas sort of anterior and posterior, caudate and dorsal, similar in the rat. So there's a striking similarity in the areal boundaries for these 4 clusters. So does this mean that rodents are a good model for human functional connectivity and human frontal cortex circuitry? Well, let's look at that in more detail by examining the connectivity of the medial frontal cortex with the rest of the brain. So David picked 7 areas that are in common to the rat, the marmoset, and the human. So this is auditory cortex, sort of A1, posterior parietal cortex, S1 and M1, motor cortices, amygdala, insula, and striatum. And he looked at the connectivity between each of these clusters and those particular areas in each animal. So you could pick more areas. We picked 7 in part because we were limited a bit by rs-fMRI coverage for animals that are in the Sphinx position, but this is certainly extensible. So this connectivity between each cluster of the medial frontal cortex and the 7 brain areas can be displayed as a fingerprint in sort of heptagonal coordinates. So that's what these are. These are fingerprints, and I'll go into more detail about them in a second. So let's look at cluster 3, for example. So this is looking at how cluster 3 in each species connects to each brain area in each species. So you can see that cluster 3 in the rat connects very strongly to primary motor area, whereas in the marmoset, in the human, cluster 3 is more distributed between insula, striatum, and auditory cortex, for example. So you can see that the fingerprints are different. And you can evaluate how these fingerprints are similar or different by using cosine similarity or any number of other metrics. That's shown here in matrix form, but the summary is that the fingerprints between humans and rodents are different for all 4 clusters. The fingerprints between marmosets and rodents were different for clusters 1 and 2. And the marmosets in general show much greater intraareal similarity to humans than rodents show to humans. So that's comparing rat and marmoset to human, okay? So let's think a little bit more about prefrontal cortex. Well, you know, even the definition and what people think of for prefrontal cortex really depends on who you ask. If you ask pharmacologists, you know, most of what they discuss about prefrontal cortex is really in terms of neurotransmitters like serotonin and dopamine. If you ask anatomists, it's based on hippocampal and medial prefrontal cortex and dorsolateral prefrontal cortex in humans, and so forth. But rodent studies in general tend to equate medial prefrontal cortex with dorsolateral prefrontal cortex in primates. So the question is that really a valid assumption? So we can look at that with our data. So in each species, we can compare the interareal connection patterns for the medial frontal cortex with those derived from the 7 functional clusters just within the marmoset lateral frontal cortex. And that actually allows us to test the hypothesis that rodent medial frontal cortex is topographically similar to primate lateral prefrontal cortex, right? Because that is the assumption people have been making. So let's take a look at this in some detail because this is a complex figure. So in the marmoset, these are the 4 medial frontal cortex clusters, C1, C2, C3, and C4. And these are their interareal connectivity similarities with the marmoset lateral frontal cortex, okay? So you can see, for example, that C3, cluster 3 in the marmoset, has an interareal connectivity that is very similar to LFC clusters 1, 2, and 3 in the marmoset. So the next thing I want you to notice is if you compare this pattern to the pattern in humans, they are topographically extraordinarily similar. Okay, so the marmoset connectivity patterns with the cortex are very similar, and those same connectivity patterns with the lateral frontal cortex are very similar to what you see in the human. While as these patterns are not really replicated at all in the rat. So this really shows that the connectivity, even though the pattern looks very similar with these 4 clusters in all 3, the cortical connectivity pattern and that shared connectivity pattern with the lateral frontal cortex is very different in the rodent. So basically, that work shows that there is remarkable similarity in the topographical organization of the medial frontal cortex if you look in detail at the cytoarchitectonics and where those clusters lie. But the medial frontal clusters of the rat brain show preferential connectivity with motor regions, so that's M1 and S1, whereas MFC clusters in the marmosets and the humans show broad cortical and subcortical connectivity. So there's a big difference in the connectivity of the MFC, even though it's sort of mesoscale topography looks the same between the 2. And so there are very clear differences between the rodent medial frontal cortex and primate lateral frontal cortex in terms of interareal connectivity. So one really cannot equate LFC or dorsolateral prefrontal cortex in primates with rodent medial frontal cortex. That's simply not valid. So this work was published just a couple of weeks ago in PNAS. So everything I've shown you so far is done with anesthetic. And anesthetics have a number of confounds. So Yuki Hori, another postdoc in the lab, has shown that not only does anesthesia change the strength of connectivity, but it changes different patterns without substantial averaging. So in the isoflurane anesthetized marmoset, you see that the default mode network, for example, is missing its frontal components. While as in just the fully awake marmoset, in the same number of runs, basically, actually fewer number of runs, you can detect the frontal cortex. So there's a strength difference, as you see in the difference, but there's also a spatial difference. So you can look at this in other ways. So if you look at thalamic connectivity in a fully awake versus an anesthetized marmoset, and you look in the cortex, so cortical connections first, that's shown in the blue. you get sort of a one-to-one mapping between the correlation or the connectivity strengths in the awake animal and in the anesthetized animal. But if you look at thalamic connectivity to the cortex in the fully awake animal and in the anesthetized animal, you can see that's actually quite skewed. So the strengths change and the patterns also change. So you have to be careful whenever you're looking at resting state, and you're looking at resting state in anesthetized animals in particular. So again, inspired by Afonso Silva's pioneering work at NIH, and it really was because he was doing awake animals 7 years ago, we decided to convert our 9.4 into an ultra-stable, ultra-high SNR sort of mesoscale imaging machine. And to do that, we upgraded to the Bricker platform with some very kick-ass gradient amplifiers. Blaine Chronick's lab here at Western designed and built an extraordinarily powerful Marmoset specific imaging gradient, which has now got a 15-centimeter diameter. So there's a lot more room for animals with chambers on them, for example. And we designed an animal holder that allows for animals with recording chambers because we want to be able to do optogenetics, chemogenetics, electrophysiology, and microstimulation in the magnet. We want to keep an eye on what the animals are doing, so we have a camera in there, and we want to be able to reward them just as we do for normal NHP experiments. So we have a sipper tube as well. And this setup, which requires an implanted animal, keeps motion generally well under 50 microns and rotations well under 1/10 of a degree. So the holder also has an RF coil built in with conductive screws that kind of complete the RF coils. And we have pins that allow us to actually center animals in case the implant, for example, is not perfectly straight, so that we can register these to atlases more efficiently. So in as little as an hour of imaging an awake animal, you can actually extract 8 or more resting state networks from a single animal. So this is quite an improvement over the anesthetized case. And this work is described in a neuroimage paper published last year as well. One of the problems with imaging animals with chambers is you get these susceptibility artifacts in any kind of T2 star weighted image like EPI. We actually used a common gel that is available, that is used surgically and otherwise. So David filled the head chamber with that gel, and that actually solves the problem in terms of signal dropout. So usually people don't show you their EPIs. You'll notice I'm very fond of it because we're very proud. So this is 500 micron isotropic fMRI of the marmoset. You can see it looks quite respectable, and you can see anatomical landmarks. That, of course, helps with registration. So in the awake marmoset, then you can look, these are 3 animals. You can look at these same networks that were originally identified by Merriam many years ago. And you see that even in the individual animals, there's remarkable similarity between the networks in these animals. It's just so much better, really. But of course, awake marmosets can do more than just lie there. They can do tasks, and that's kind of the whole point of being able to do awake behaving animals in a scanner. To do the first of the tasks, we had to actually do a few more things. So we took an old projector, bounced it off a first surface mirror that's located right around here, and onto a screen that's right at the bore, front of the bore of the magnet. And then our programming geniuses figured out a way to do real-time reconstruction of the EPI images coming off the scanner, as well as real-time motion tracking of those images, so that we can keep an eye on what the animal is doing. We can stop a run if they're misbehaving or often falling asleep. The eye tracker is useful for that too. So this allows us now to start to do tasks in the magnet. And the first task we did was quite simple to map out visuo-saccadic areas in the marmoset. So this is a block design paradigm where we have fixation, a pseudo-fixation, a block where we have these movie trailers that are 1.5 by 2 1/4 degrees roughly, presented in even and odd blocks. Even blocks are very close to the fixation point, odd blocks are further away. We do this, there are 8 task blocks, roughly 4.5 minutes, and we can repeat this a number of times, of course, basically till the animal decides he wants to fall asleep. So these are what the maps look like. You have to jump through some hoops, I should mention, because we reward the animal just before the start of the visuo-saccadic task and right at the end if they hold still. And so there are licking confounds in this. So we can use kind of a hybrid ICA approach to actually separate these two. And what you see here is these are 3 marmosets, 3 separate runs. These are the visual maps, and these are the reward or licking maps. And you can see they are extraordinarily good. Any single subject is comparable to any other single subject. And this is the real benefit of doing the awake animals. Of course, you can put this on the NIH Atlas that Afonso's group published a couple of years ago. So now you have all the boundaries. So these are from 3 animals only with 6 runs. So this is the visual task, and this is the licking reward task. We made sure the sipper task is not in the mouth, so the animal actually has to extend its tongue. So there's a distinct motor movement here. And so you can see that the visual task is activating V1, V2, V3, LIP areas. It extends down into the temporal pole, as well as we have these frontal cortex areas. So of course we can do the same thing in humans. So we did in our 70, minus the licking for juice rewards. We just pay our subjects instead. So we use Avitech rear projection, and we did exactly the same task. And these are the maps from that. We only needed 2 subjects and 3 runs. We have a super duper stable 7T as well. So it doesn't take much to get extremely stable map. And you can compare these side by side. So this is 3 marmosets and 2 humans. And as you can see, both on the medial surface as well as on the lateral surfaces, there is remarkable concordance between the maps. So one issue in marmosets is there really isn't a well-characterized map of the frontal eye fields in marmosets, surprisingly. And so we also did resting state in these same animals. And we know that there's connectivity between area MT and the frontal eye fields, as well as the superior colliculus and the frontal eye fields. So all we did was drop a seed in MT and drop a seed in superior colliculus. And both of those generate an area out here in the frontal cortex that coincides very well with the task-based fMRI frontal cortex activation. Okay, so that there's convergent evidence that this map reflects some degree of eye movement activity in the frontal cortex, as well as all these visual areas. Okay, so then we started to look at some more details about this frontal area in particular. So old world primate species, as well as us, share sort of a common architecture for face processing circuitry. So along the occipitotemporal axis, there are multiple face selective patches in both humans and non-human and macaques, certainly. And these are functionally connected with larger face processing network area that includes subcortical areas like hippocampus and amygdala, as well as face selective patches in frontal cortex. Now, if you take a free unhead fixed marmoset and you let them look at faces, what you see is they use head movements to orient to the faces, but the red trajectories that you see are the eye movements as they explore the faces. And this suggests that, there may be circuitry in the marmoset that is actually supporting things like social cognition, that recognition of what's going on in the face, and is it friend or foe, and so forth. So there is evidence of this in the macaque monkey. So Winrich Frewald at Rockefeller showed just a couple of years ago that there is a face-sensitive patch in the frontal cortex. And he tried to sort of separate this into areas that are involved in terms of a direct gaze, where the animal is looking at another animal or another face staring straight at it, versus a side view of the head, for example. The control condition is usually scrambled faces. So David developed a paradigm to look at this in the marmoset. Again, it involves fixation, directed gaze at pictures of real marmosets in our labs that are looking straight ahead in a block design, scrambled versions of those, and then averted gaze versions of those. So we used 10 animals in this study, just 4 needed for awake fMRI, another 5 for resting state fMRI, and 5 for some eye tracking that I'm not going to talk about, but people may ask. And so these are what the face masks look like. So these are both the averted and direct face activations compared to fixation. These are what the scrambled faces look like, and this is what the difference looks like. And you notice, in addition to all these visual areas along the occipitotemporal axis, there's also activation in area 45 and 47 here, as well as subcortically. So we'll come back to that. So you can also look at the directed versus averted gaze maps. And if you do that, what happens is you see a less broad map, so still large areas, but now islands of activity in MT and FST and TE, as well as activity in frontal areas, both on the medial bank as well as the lateral bank. And so we actually think, we hypothesize that this cluster in 8AV 45-47 is actually related to small eye movements, because we know where FEF is in these animals, and that's actually these activity maps are too far ventrolateral to be related to eye movement. So it's likely that they're face specific. And this patch in particular here seems to be specific to the directed gaze condition. So it's a candidate for socially relevant face processing. And so we can compare what we found in our face-sensitive patches with what actually Afonso's group looked at a number of years ago using a very similar paradigm, but they did not find this frontal area. But we are completely concordant in terms of the other areas. And it's also the areas we find in the occipitotemporal pole, as well as the frontal cortex, are completely concordant with what's been seen in macaques. So that's pretty nice. It shows that, the social and face processing circuitry in the marmoset actually echoes what we see in the macaques, and what we've also known from fMRI is seen in the humans. And talk about waking up to good news. This paper was actually accepted in Nature Communication this morning, David, so congratulations. And Stefan says the $4,000 publication charge check is in the mail. So there are other ways to look at this concordance. So using these known, looking at the patches in the occipitotemporal cortex, we can seed the resting state fMRI, and we can look at where that connects. And we also get this very robust connectivity, particularly to the medial bank here of the frontal cortex. And we can compare injections, tracer injections made in these areas, 8B24, with tracer data that has just been published, also in Nature Communication, in marmosetbrain.org. So there are 2 injections that have been made in this area, 8B and 24C. And indeed, those tracer connections also map very nicely to what we see both with the resting state and the task-based fMRI. So there's a concordance of methodologies here. So what I showed you was really the idea of mimicking what Vinrich did in the macaque and what Afonso did in the marmoset in terms of a task. But you can go beyond that. So we built a second holder, and we just put it in the 9.4T along with an LCD screen here that can be made clear or translucent. And we put a second marmoset with this holder into the magnet, and we basically redid the whole paradigm with the marmoset that we were imaging staring either at a translucent screen or making it transparent so they could see a real animal whose head is moving around. And this is done with the pictures, and this is done with the real life marmoset. And so you can see that there's a lot more visual activity, slightly different. This could just be because where the visual fields are, but it's a very strong activity going down into the lateral occipital sulcus as well. And there's a very strong locus of activity, exactly the same place that we saw when we presented pictures. So this is really cool. So it doesn't matter whether you're showing a marmoset, a picture of a marmoset, or an actual marmoset's face. So this is like better than virtual reality. It's reality. And the nice thing is you see convergence. And you also start to see other areas in very much, this is in cluster 6 of our cluster analysis. As well as in cluster 4. So those can be explored further. And then of course, if you can do something like this, then wouldn't it be cool if you could actually image both marmoset brains at the same time? So unfortunately, we can't actually do that in the 9.4T because the homogeneous region of the gradient coil isn't large enough. And at 9.4T, you would really never be able to shim the magnetic field homogeneously over both brains at once. But Kyle Gilbert and his group are geniuses. And so our RF guys have built a coil for the Prisma. And that's basically 2 copies of what we use in the 9.4T. One plugs into one of the head coil plugs, and one plugs into one of the body coil plugs, essentially. So now we have 2 marmosets who can be separated in distance, but of course the gradient's homogeneous region in Prisma is very much larger than it is in the 9.4T. So we can look at animals that are very close, animals that are very far, they can smell each other or not. We can do all kinds of things in terms of looking at social activities, and we can image both brains at the same time. We can't do it at the same spatial resolution, obviously, as in the 9.4T, but we think we might be able to get 1 to 1.5 millimeters. So at least at the macro scale, this will be a way of simultaneously imaging two animals at once and looking at their social interactions. So the last thing I want to talk about is the idea, again, of exploring human and marmoset homologies. So I talked in length about how we've developed all these tools to image awake behaving marmosets in the 9.4T. Well, we've been doing the same thing in our 7T. So we have been using our own 8-channel transmit coil and 32-channel receive coil for many years. This is a new version of that coil, which uses dipoles instead of loops for the transmit. What that actually allows us to do is have this massive wide-angle visual field, which vision neuroscientists love because you can get like 40 degrees of visual angle here. So you can do things that really were not possible with fMRI for much of the past 30 years almost. Plus you can use eye trackers. So we have a binocular eye tracker that works in the 7T, and we have an Avitech projector, and these both work basically to present images and tell us where on the images subjects are looking. And of course, we can do the same thing because we can do eye tracking in the scanner in the 9.4, because there's the camera there. And so what we've just started to do now is actually show movies of marmosets doing things, as it turns out, to both marmosets and humans. Exactly the same movie, 15 minutes long, in each of the scanners. We do it just once because we have a lot of signal, and we can now start to compare how marmosets are processing these complex scenes and storylines, and humans are processing the same things, and look for the homologies in those. So that's quite exciting. I was a volunteer for this about a week ago, and hoping I outperformed the marmoset in terms of the storyline, but we'll see what happens. Stefan Everling is currently analyzing that data. So in conclusion, I just want to say that, whatever translational model you want to use requires careful validation with the homologies that are relevant to the problem you're looking at. Those could be at the molecular or the cellular or the receptor or circuit level, ideally all of those, so that you can look at pharmacological interventions, for example, that are relevant between your model and a human. MRI is a super powerful tool to establish both macroscopic and mesoscopic homologies between model systems and humans. And marmosets in particular seem to be a viable preclinical model for not only just studying the frontal and prefrontal cortex and social activities of marmosets, but as models for disorders that involve those areas. And just a reminder that these areas are considerably more developed in the marmoset compared to the rodent, and similarly organized to what we've seen in the macaque. So they're a nice intermediate model for doing things. With that, I'd like to thank the people who've poured millions of dollars into our lab for supporting various aspects of what I've talked about, and happy to take questions. So thank you very much.